Hello students, welcome to my class. Today we'll discuss very different and very interesting topic regarding the genes. Exactly, if someone asks you what is a gene, then how will you define it? Gene, very easily it can be defined as that it is a functional part of a DNA which codes for a functional protein product. It is the functional, structural and functional unit of DNA. Gene is called. Okay, so different types of genes. It is not the particular definition of a gene that if a piece of DNA which codes for a protein, it is called gene. It is not the particular definition of the gene. Exactly, many different types of genes are available in case of the eukaryotic system, and not all the genes are protein coding genes or cistrons. Okay, so what are the types of genes present in eukaryotic system? What are the functions of all these different types of genes? Let's discuss. Okay, see here, what is a gene? Just as we have discussed, gene, normally we can define it. It is the structural and functional unit of DNA which codes for a functional protein product. But there are different types of genes present. See, first is the housekeeping or constitutive genes. Housekeeping genes are there. What are housekeeping genes? These housekeeping genes, they continuously express, they continuously remain switched on, and their protein product, it is continuously being synthesized inside the cellular environment because their protein product is needed continuously inside the cell. Supposed to be, we can give examples of any of the normal metabolic pathway. Supposed to be it is about glycolysis, or it is about the Krebs cycle. Means these metabolic pathways, they run throughout our life each day, and the proteins or the enzymes which are involved for these pathways, these are continuously being synthesized by the genes which code their products. So these genes, they remain continuously switched on and they continuously express. These continuously expressing genes are called housekeeping genes. Housekeeping genes never switch off. Housekeeping genes are also known as constitutive genes. These genes, they continuously synthesize their protein product which is needed for the cellular environment. Second, come to the non-constitutive genes. The non-constitutive genes are luxurious genes. These genes, they synthesize their product, they are expressed as per the need of the cell. And these non-constitutive genes or luxurious genes are of two different types. See here, first is inducible gene and second is repressible genes. Inducible genes means what type of genes are called inducible genes? These genes, they are induced by the presence of a particular substance called inducer, and basing upon the presence or absence of this substance, these genes either they are switched on or switched off. These are inducible genes. Okay, what are repressible genes? Repressible genes means what? These genes, they continuously synthesize their protein product. They are continuously expressed, but when the protein product is more, when their own product is more for the demand of the cell, then automatically the end product of this gene, expression of this gene, the end product of the protein itself, it acts as a repressor and it represses the gene. It suppresses the gene's expression. So these are called repressible genes. So mostly they remain switched on, but when the protein product which is synthesized by those genes is more than the demand of the cell, then these genes, they switch off automatically by the end product of the expression. Okay, then come to the third, multi-genes. What are these multi-genes? These are the group of similar genes. These are the group of similar genes, they express continuously for fulfilling the requirement of the cell. Depending upon a particular protein product, they are continuously working. These multi-genes, these are homologous genes, and they were involved for a particular pathway, and they synthesize the product as per the demand of the cellular environment. These multi-genes. Okay. Then the fourth, pseudogenes. What are these pseudogenes? Pseudogenes are homologous to functional genes. See here what I have written here. Pseudogenes are homologous to functional genes. Pseudo means false, as all of you know. These pseudogenes means these are false genes. They appear like the normal gene, but these genes, they do not express and they do not produce a functional protein product. Why? Because of any insertion or deletion mutation, maybe, or because of what? Because of inactivation of the promoter region, okay, or because of presence of any non-coding sequence inside the gene. Because of that, those pseudo genes, these are homologous to the normal functional genes, but they can't produce their functional product. These are pseudo genes. Then fifth is split genes. What are these split genes? As per the name indicates that split means there is splitting of the coding sequences, and these genes are also known as interrupted genes and with intervening sequences. These split genes, they are having intervening sequences which do not code for a protein, okay, and because of the presence of such intervening sequences inside the gene, these genes also do not produce functional protein product. Then come to the transposons or jumping genes. It's a very typical peculiar thing present in our chromosomes, that they are having transposons or jumping genes. These jumping genes, they are mostly present toward the telomeric region of the chromosome. These transposons or jumping genes, they are having repetitive DNA sequences. They are having repetitive DNA sequences, either similar or inverted repetitive DNA sequences are present. These jumping genes or transposons, they do not code for any protein, but they can jump, they can change their position, they can discontinue from a particular part of the chromosome, from the end piece of the chromosome, and they can ligate themselves to another chromosome, okay. As they jump inside the nucleus, inside the chromosome constituency, these are called jumping genes or transposons. Then overlapping genes. These overlapping genes are not found in higher organisms, higher vertebrates. These are found in case of bacteria and animal viruses. These overlapping genes which code for more than one polypeptide. These genes are called overlapping because they do not code for a particular protein product. They code for more than one proteins, and these genes are called overlapping genes, okay. Next, regulator gene, operator gene and promoter gene. These three are called regulatory sequences. These three, regulator gene, operator gene and promoter gene, these are present upstream of any of those cistrons, okay. So the regulator gene, operator gene and promoter gene, they regulate the expression of a particular cistron, a protein coding gene, okay. So come to the structural gene or cistron first. What are the structural genes or cistrons? Cistrons are the protein coding genes present in the chromosome. Not all the genes present inside the DNA or chromosomes, they code for the protein. Only the protein coding genes are called cistrons. They are having coding sequences, that is exons, and non-coding sequences, that are what? Introns. Repeatedly. Alternatively, the exons and introns are present inside the cistrons, and cistrons, they code for a particular protein product. So the structural gene or cistron which codes for a particular morphological or functional character. It codes for a particular morphological or functional character, producing a particular functional protein product. And these cistron or structural genes, they are under the control of regulator gene, operator gene and promoter gene, because these three genes are present upstream to the cistron. In one of my classes also, in the previous classes, I have also discussed all these things, that in case of eukaryotes, monocistronic mRNAs are produced. Monocistronic mRNA means what? A single cistron or single functional gene produces a single mRNA, because each cistron is preceded by these three regulatory sequences: regulator gene, operator gene and promoter gene. What are these genes? Come to the regulator gene first. Regulator genes, they code for repressor protein. These regulator genes, they come under the housekeeping gene or constitutive gene part, because these regulator genes which are present upstream to the cistron's coding sequence or structural gene part, these regulator genes, they code for a particular repressor protein. They produce a repressor mRNA. The repressor mRNA, it translates a particular repressor protein. The repressor protein, it binds to the operator region which is present next to the regulator gene in a cistron, okay. So the repressor protein, if it binds with the operator gene, then automatically the cistron will remain switched off. The cistron will not express, okay. So this gene is called regulator gene, as it regulates whether the cistron will be expressed or not. Then come to the operator. Operator just acts as a switch. if the repressor protein produced by the regulator gene, it binds in the operator zone, then it remains switched off. The operator gene, it remains switched off, and it doesn't allow the RNA polymerase enzyme to move forward and transcribe the structural gene of cistron. Okay. So this operator gene, it acts as a switch to turn on or turn off the transcription of the cistron. Then the promoter gene. What is the promoter gene? This promoter gene, these must be present and this must be active in case of cistrons. Neither the cistrons can't express. These promoter genes means these are the site of the DNA upstream to the cistrons structural genes. The promoter gene is present, and the promoter gene, it provides the site for the binding of RNA polymerase enzyme. Particular RNA polymerase enzyme, it binds to the promoter region of the cistron so that it can scan the structural genes nucleotide sequence and can transcribe the mRNA. Okay, so promoter gene is the site for the RNA polymerase binding. Then come to the terminator gene. Terminator gene, this is the DNA part or DNA piece which is present towards the downstream of the cistron. Towards the downstream of the cistron, towards the active downstream of the cistron, the terminator gene is present where transcription terminates, where RNA polymerase enzyme dissociates from DNA and automatically the transcription process is terminated. So DNA site, this is the DNA site where RNA polymerase activity stops and transcription terminates. Okay, so this is about terminator gene. Then come to silencer gene. What are silencer genes? These genes, they don't remain silenced. Silencer means they silence other activities, mean the other genes activities. These silencer genes, they silence other genes. If the protein product of a particular gene is not needed by the cell, then that particular gene will be silenced by another gene, and this another gene which silences is called silencer gene. Okay. Then enhancer gene. Enhancer genes, what? They enhance other genes activity to produce a particular protein product if it is needed more inside the cell. Okay, enhancer gene. Then integrator gene. This is very important, that integrator genes are present in case of eukaryotic systems which integrate, and integrately they enhance other genes which are involved for a particular metabolic pathway. And these genes may be present in different chromosomes or different parts of a single chromosome also. All these genes at a time, they will be activated by these integrator genes. Supposed to be a particular biochemical pathway which involves 10 or more than 10 number of enzymes. So it must be involving more than 10 number of proteins. So it must be involving more than 10 number of cistrons. So all these cistrons or protein coding genes, whether they are present in the single chromosome or in different parts of the chromosome or in different chromosomes, all these genes will be switched on at a time when the metabolic pathway is going to be there, occurring inside the cell. Okay, so these integrator genes, they are responsible, they integrate the group of genes, they integrately activate the group of genes which are involved in a particular metabolic pathway. So these are different types of genes present in eukaryotic chromosomes, their functions and how they regulate the cistrons activity. Cistrons are the protein coding genes, and very less genes present in our chromosomes are cistrons. Only 2% or less than 2% of the genes present in our chromosome, they code for proteins. Rest of the genes, they do not code, and they are having such activities. And another thing to tell you also, some genes are there which do not code for protein. They do not transcribe mRNA, but they transcribe either tRNA or rRNA. These genes are also present which help for transcription. Okay, so these are all about the genes, their functions, and this is a very interesting thing, that in case of eukaryotic chromosome, in case of eukaryotic system, such type of genes are present with different types of functions to regulate the gene expression in eukaryotes. Thank you for watching. Share and subscribe my channel for upcoming videos. Thank you.